When you think of, forest architecture, you naturally just look up.
Right, yeah. You look at the canopy.
Exactly. You look at the towering canopy of a subarctic spruce or the massive trunk, the branches, and you assume you are looking at the builder, like the dominant force.
That's absolutely.
But if you step off the established trail and into a damp, freezing boreal bog, that entire architectural model just flips completely upside down.
It really does.
Because the true architects of that ecosystem are actually hiding right beneath your boots, and they are completely covered in slime.
Which is just wild. I mean, it forces a complete recalibration of how we view structural dominance in nature.
Yeah.
We're just so conditioned to associate size and verticality with, ecological importance.
Right. The bigger the tree, the more important it is.
Exactly. But in these harsh northern temperate zones, the actual foundational engineering, the nutrient distribution, the chemical defense systems, even the regulation of the soil's thermal properties, all of that is being managed by organisms that barely breach the surface of the moss.
Which brings us to the actual subject of our deep dive today. So for everyone listening, today we are looking at a single, unassuming, grayish-purple mushroom,
Yes.
Lactarius trivialis, commonly known as the Slimy Lead Lactarius or the northern milkcap.
It's a fantastic subject.
It is.
Yeah.
And we have a massive stack of sources to get through today. I mean, we're jumping from 19th century taxonomic texts all the way to some cutting-edge 2025 soil ecology research.
The range of data on this one organism is just staggering.
Yeah, so our mission today is to synthesize all of this and figure out how a simple waterlogged fungus essentially dictates the terms of survival in the subarctic.
And what's fascinating here is that it's a remarkable organism to be traced through the scientific literature. I mean, if you were to just glance at it in a finished bog, you'd likely dismiss it.
Just a slimy gray lump.
Pretty much.
Yeah.
But the deeper you look at its physical engineering, its highly aggressive chemical weaponry, and ultimately its role in global carbon cycling, you realize it is running the entire show.
It really is. And to really understand how advanced this organism is, we need to look at how human science first tried to categorize it.
Right.
Because long before we were looking at its effect on global carbon sinks, we were just trying to figure out what it was. And that takes us back to 1838.
Specifically to the work of Elias Magnus Fries.
Right.
He was this monumental figure in Swedish mycology. And 1838 is the year he published his defining work, Epikrisis Systematis Mycologici.
Which is a very cool title.
It is. But context is everything here,
right?
Because Fries is operating in an era completely devoid of genetic sequencing.
Oh, yeah. No DNA analysis, no molecular phylogenetics, none of that.
Nothing. So he is tasked with organizing the sprawling, chaotic diversity of the fungal kingdom using only what we call macromorphological characters.
Okay, let's unpack this. So he's relying entirely on macroscopic features. He's categorizing this immensely complex kingdom of life based strictly on what he can see, touch, and taste out in the field.
Exactly. He had to look for reliable, observable physical traits that hinted at a deeper biological relationship.
Right.
And for this particular group of fungi, the defining trait was the production of a milky fluid.
The milk.
Yeah. If you cut the gills or snap the stem of one of these mushrooms, it aggressively bleeds a liquid latex.
It's so weird.
It really is. And this is why Fries placed them in a genus he named Lactarius, which is derived directly from lactus, the Latin word for milk.
But he didn't just stop at, it bleeds milk, right? He drilled down further into the exact type of milk because he placed Lactarius trivialis into a specific subgenus called piperites.
He did, yes. And the piperites group is characterized by robust, fleshy fruiting bodies, but more importantly by an intensely acrid, peppery taste.
Peppery.
Extremely peppery. Fries recognized that this burning sensation wasn't just some random flavor profile. It was a consistent defensive characteristic shared by a specific lineage of these milk caps.
So what you're saying is he was essentially using his own taste buds as a crude biochemical assay.
Basically, yes.
I mean, he didn't know the molecular structure of the toxins, but he just knew that a group of mushrooms all producing the same burning sensation must be closely related.
That is precisely what he was doing. And honestly, it is a testament to his observational brilliance.
It's fascinating, but I mean, from a modern scientific standpoint, it seems incredibly precarious.
Oh, for sure.
Grouping organisms by how peppery they taste or whether they leak milk. It's kind of like an early automotive engineer categorizing cars by what color fluid they leak onto the driveway, rather than looking under the hood at the actual engine architecture.
That is a very sharp way to frame it.
Just feels like observing the exhaust rather than the
He
engine.
was absolutely categorizing by the metabolic exhaust.
Mm.
But the striking thing about Fries' taxonomy is how incredibly durable it turned out to be.
Wait, really?
Yeah. When modern scientists finally did look under the hood using genomic sequencing, they found that Fries' morphological groupings, the ones based on milk and pepper, largely aligned with the actual genetic clades.
That is wild.
Isn't it? The macroscopic features he identified were actually highly accurate proxies for deep genetic divergence. He managed to identify a highly conserved evolutionary strategy.
Wow. Well, let's talk about the physical manifestation of that strategy. Because if you are walking through a boreal bog, the environment itself is actively trying to destroy anything fleshy and nutrient-rich.
It's a very harsh place.
And you mentioned the cap earlier. We're talking about a substantial structure here, like 6 to 20 centimeters across. And it has a very specific color progression, right?
Yes, the morphology shifts quite dramatically as it ages. In its juvenile stage, the cap possesses this deep, striking, purplish violet or leaden gray coloration.
Which is where we get the name, the Slimy Lead Lactarius.
Exactly. But as the fruiting body matures and the cap expands, that deep lead color fades outward. It leaves a brownish gray or sometimes a pinkish beige hue, often presenting with faint concentric zones or darker mottled blotches across the surface.
That modeling has to be an adaptation for camouflage.
Oh, definitely.
Because if you look at the floor of a subarctic forest, it is just this chaotic mix of shadows, dead spruce needles, sphagnum moss, decaying birch leaves. That grayish, zoned pattern would break up the visual outline of the mushroom perfectly against the leaf litter.
Spot on. It blends right in.
But the camouflage isn't even the most prominent physical feature. The defining characteristic is the mucus.
Ah, yes. The slime.
Yeah.
It is an incredibly viscid mushroom. When the environment is damp, the entire cap is coated in a thick, highly tenacious layer of mucilage.
Which, from a human perspective, is viscerally unpleasant to touch. I mean, it's just gross.
And it's not a fun texture.
Right. But evolutionary biology doesn't invest metabolic energy into producing thick layers of slime just to be unpleasant to hikers. There has to be a rigid biomechanical utility to that mucus layer.
There is a dual utility, actually.
Right.
The first is moisture regulation. The subarctic summer is relatively short, but it can be subjected to periods of intense, dry, scouring winds.
Okay, so it gets dry quickly.
Very quickly. And a mushroom is fundamentally a hydraulic system. It relies on turgor pressure, water pressure inside its cells to maintain its shape and forcibly eject its spores.
So if it dries out?
If the dry wind strips the moisture from the cap, the mushroom desiccates. The reproductive cycle completely fails. So the viscid layer acts as a highly effective hydrophobic barrier.
Oh, so it seals the water in.
Exactly. It seals the internal moisture inside the fleshy tissue, completely buffering the organism against sudden drops in ambient humidity.
It's essentially wearing a customized biological raincoat. But instead of keeping the rain out, it's keeping its own internal hydration locked in.
That's a perfect way to put it.
But you mentioned a dual utility. What is the second function of the slime?
Mechanical defense?
Really? Slime as armor?
Basically, yeah. Because the subarctic bog is teeming with micropredators, nematodes, tiny fungivorous insects, larvae, all looking for a nutrient-dense meal.
Right, because mushrooms are packed with calories.
Exactly. And that thick mucus layer acts as a highly viscous physical trap.
Like flypaper.
Yes. Small invertebrates that land on the cap in an attempt to bore into the flesh find themselves bogged down and immobilized in the slime before they can even reach the cellular structure of the mushroom.
Wow, so the cap is the sealed, camouflage, flypaper-coated vault.
Pretty much.
But a vault is useless if it sinks into the mud. And this brings us to what I honestly consider the most elegant piece of physical engineering on this entire organism, the stem.
Ah, the Hopperuscu.
Yes. If you look at the Finnish sources, they revere this mushroom. They call it the Hopperuscu. And the absolute gold standard identifier for a true Hopperuscu is that the stem is 100% hollow.
Completely hollow.
It is a perfect empty cylinder.
And that hollow stem is a masterclass in biomechanical optimization. You really have to consider the specific soil mechanics of its habitat to appreciate it.
Right, because it's in a bog.
Yes. Lactarius trivialis does not grow on solid, compact earth. It thrives in highly saturated waterlogged soils, usually submerged deep within wet, spongy layers of sphagnum moss under coniferous canopies.
And if an organism builds a solid, dense, fleshy stem in that environment, it is facing two immediate physical crises.
Exactly. The first is sheer weight. A solid stalk composed of densely packed mycelial tissue is heavy.
Right.
In a semi-liquid bog, that weight causes the organism to slowly sink back into the muck, defeating the entire purpose of elevating the cap to catch the wind for spore dispersal.
Because if the cap is on the ground, the spores go nowhere.
Right. And the second crisis is the anoxic nature of the bog. Waterlogged soil lacks oxygen.
Okay, so it can't breathe down there.
Exactly. A dense, fleshy stem submerged in anoxic water acts like a sponge. It absorbs the surrounding moisture and succumbs to bacterial rot incredibly quickly.
So by hollowing out the stem, the fungus solves both problems simultaneously.
It does.
It actually reminds me of the engineering principles behind carbon fiber tubing used in modern aerospace or, like, drone manufacturing.
Oh, that's a great comparison.
Yeah, because if you need a structural support that can withstand horizontal wind shear, a solid cylinder is incredibly inefficient. It just wastes mass. In physics, the resistance to bending is maximized when the material is pushed as far away from the central axis as possible. So by building a hollow tube, the mushroom maximizes its second moment of area. It achieves the exact same structural rigidity as a solid stem, but at a fraction of the weight and a fraction of the metabolic cost.
That is a perfect biomechanical translation. It minimizes the carbon investment required to do the structure, ensures the stem remains buoyant and lightweight in the saturated moss, and still provides the vital rigidity needed to rapidly elevate the cap into the air currents.
So the physical architecture is just perfectly dialed in. It has a hollow, lightweight composite stem keeping it elevated above the rod. It has a mucilage-coated, moisture-locked cap preventing desiccation.
Yes.
But as brilliant as that physical fortress is, it only protects against the environment and microscopic pests.
Right. The small stuff.
Exactly. The cap is still a massive, water-filled, nutrient-dense package of calories sitting in a forest full of starving mammals and larger insects. A hollow stem won't stop a squirrel.
No, it certainly will not.
So to survive macro predators, it relies on a defense mechanism that is, frankly, terrifying in its efficiency.
You are talking about the chemical landmine.
Yes.
And this brings us back to the milky latex that Elias Magnus Fries observed back in 1838. We now understand the precise molecular machinery behind that milk, and it is a marvel of evolutionary biochemistry.
Okay, let's get into it.
So the latex is stored under high pressure within a specialized network of internal plumbing called lactifers, which weave through the flesh and gills of the mushroom.
And when the mushroom is whole and undisturbed, the milk is bright white.
Right.
But the diagnostic feature is what happens when that milk is exposed to the air.
Correct. When the tissue is damaged, the pressurized latex violently bleeds out. Upon exposure to ambient oxygen, specific compounds within the latex rapidly oxidize, causing the bright white milk to dry into a very distinct grayish-green crust right on the wound site.
The color change is a great visual indicator for foragers, but the actual defensive mechanism isn't the color itself.
No, not at all.
The defense is the invisible chemical reaction happening at the exact same moment. The sources refer to this as the velutinal ester pathway. This is basically a binary weapon system.
Binary weapon is exactly the right terminology. The ingenuity of this system lies in compartmentalization.
Okay.
When the Lactarius trivialis is intact, the lactifers contain a vast reservoir of highly stable, completely inactive precursor molecules. These are specialized fatty acid esters, the primary one being a molecule called stearoylvelutinal.
Let me stop you there just to make sure we are tracking the chemistry for everyone listening. The stearoylvelutinal is essentially a prodrug. It is totally harmless in its current state.
Completely harmless.
Yeah.
It is a large, bulky, stable lipid. The mushroom can store massive quantities of it without damaging its own tissues.
Right.
However, stored in completely separate cellular compartments adjacent to the lactifers are highly reactive enzymes, specifically esterases and oxidases.
Ah, okay.
And as long as the mushroom's cellular architecture remains intact, the inert precursor and the reactive enzymes never meet.
Oh, I see. It's like the two separate chambers inside an industrial glow stick.
Yes.
The chemicals are totally inert until you bend the plastic, shatter the internal glass vial, and force them to mix. The snap of the glow stick is the bite of the predator.
That is an excellent analogy, yes.
Yeah.
The moment a mammal or an insect bites into the cap, the physical trauma ruptures the cellular walls, violently mixing the compartments.
And then boom.
Boom. Within a fraction of a second, the esterase enzymes collide with the stearoylvelutinal. A massive enzymatic cascade initiates. The enzymes hydrolyze the stable ester, physically cleaving the molecule apart, and then the oxidases rapidly rebuild the fragments into entirely new, highly toxic compounds.
And what are the resulting compounds from that enzymatic explosion?
The reaction yields stearic acid, which is a basic fatty acid, and two highly reactive sesquiterpene dialdehydes known as isovelleral and velleral.
Okay, so those dialdehydes are the payload. But if we think about this from a strategic evolutionary perspective, I have to question the timing.
How so?
Well, if the enzymatic cascade is only triggered by the physical destruction of the cells, meaning the predator has already bitten into the mushroom, hasn't the defense system already failed?
I see what you mean.
The organism has already sustained catastrophic structural damage. It's already been chewed on.
It seems counterintuitive until you analyze the speed and the biological targets of the dialdehydes. The system is designed to sacrifice a fraction of a millimeter of tissue to save the entire reproductive organ.
Okay, walk me through that.
The dialdehydes interact directly with the TRPA1 pain receptors in mammalian tissue. The moment those newly synthesized molecules hit the tongue or the mucosal lining of the predator's mouth, they trigger an immediate, intensely painful burning sensation.
Wow.
It is not a delayed stomachache. It is instant, searing chemical heat.
So what you're saying is the predator doesn't even finish the first bite. The pain profile is so aggressive that the animal recoils, drops the mushroom, and permanently associates that specific grayish-purple visual profile with intense pain. It basically prevents the second bite, which is the one that would actually destroy the spore-producing gills.
Exactly. It achieves immediate behavioral deterrence. But the brilliance of the velutinal ester pathway is that it is fundamentally a multi-tool. Deterring the mammal is really only the first function.
There's more.
Oh, yes. The dialdehydes, the isovelleral and velleral, are deeply cytotoxic and strongly antimicrobial.
Oh, this makes perfect sense because the bite is an open wound.
Right.
And in a damp, spore-filled bog, an open wound on a sugar-rich, fleshy mushroom is a massive liability. Opportunistic bacteria, molds, and parasitic fungi would colonize that exposed tissue in hours, causing the entire cap to rot before it could release its own spores.
Precisely. The dialdehydes instantly flood the wound site. Because they are highly antimicrobial, they essentially act as a biological cauterization agent.
Wow, biological cauterization.
Yeah, they sterilize the bite mark, kill any invading spores or bacteria deposited by the predator's saliva, and form a chemical seal over the exposed tissue.
That is unreal. It is a chemical deterrent and a broad-spectrum antibiotic synthesized on demand from a single drop of milk.
It's incredibly efficient.
But wait, whenever you have a massive enzymatic reaction like that, ripping apart molecules and rapidly oxidizing them, you generate collateral damage.
You do.
The chemical reaction itself produces massive amounts of oxidative stress, like free radicals that could theoretically destroy the healthy fungal tissue surrounding the wound, right?
Yes, and you are identifying the exact reason why this pathway is so difficult to evolve.
Yeah.
You cannot deploy a localized chemical bomb without shielding your own surrounding infrastructure.
Right, so how does it survive its own bomb?
Lactarius trivialis manages this through a secondary chemical buffering system. Its tissues naturally hyperaccumulate organic acids, primarily oxalic and citric acid.
Ah, the organic acids act as the shock absorbers?
Exactly. They act as potent antioxidants. They flood the reaction zone, scavenging the free radicals produced by the rapid oxidation of the dialdehydes, stabilizing the healthy cells immediately adjacent to the wound site.
So it contains the blast radius.
Exactly, ensuring that the necrotic zone, the area of dead tissue, is strictly confined to the actual physical bite mark.
So we have an organism elevated on a biomechanically perfect hollow stem, coated in an anti-oxidation moisture barrier, packing a binary chemical weapon that instantly synthesizes burning pain receptors, sterilizes wounds, and deploys antioxidant shock absorbers to prevent internal collateral damage.
It is an absolute fortress.
It really is. But there's an environmental variable we haven't fully addressed yet. This fortress is built in the Arctic. We are talking about regions in Scandinavia, Alaska, and the deep Baltic states. The ambient temperatures drop well below zero.
It gets incredibly cold.
Right. And fleshy fungi are overwhelmingly composed of water. From a pure physics standpoint, how does a biological water balloon survive a deep, sudden freeze without shattering?
To understand how it survives, we really have to look at the specific mechanics of freezing at the cellular level. This is the domain of cryobiology.
Okay.
For a typical organism, freezing temperatures are lethal, not because of the cold itself, but because of what happens to water when it undergoes a phase change.
Right, the transition from liquid to solid. When water molecules slow down enough, they align into a rigid crystalline lattice, ice.
Yes.
And because of the hydrogen bonds, that lattice actually takes up more physical space than liquid water.
It expands.
Oh.
And those microscopic ice crystals are incredibly sharp.
Oh, right.
If water freezes inside the cytoplasm of a living cell, those jagged crystals expand outward, physically piercing the delicate internal organelles and ultimately rupturing the outer cell membrane.
So when the tissue eventually thaws, the internal contents simply leak out. The cell's destroyed.
Exactly. To prevent this, Lactarius trivialis engages in a highly aggressive form of osmoregulation, utilizing specialized cryoprotectant molecules.
And the primary molecule highlighted in the literature is trehalose, right?
That's the one.
It's a disaccharide, essentially a double sugar. We see trehalose pop up constantly in extreme survival biology. I mean, tardigrades use it, certain desert plants use it to survive decades without water. What exactly is the trehalose doing inside the fungal cells as the temperature drops in the bog?
It is fundamentally rewriting the physical properties of the internal water. As the subarctic temperature plummets toward freezing, the metabolic machinery of the fungus detects the thermal shift and massively upregulates the synthesis of trehalose.
How massive are we talking?
In cold-adapted species, we see the internal concentration of trehalose spike from trace amounts like less than 1% of the organism's dry weight to over 20% in a matter of hours.
Wow, that is a staggering metabolic investment. It is just flooding its entire circulatory system with sugar?
Because trehalose possesses exceptional hydrogen bonding capabilities, it acts as a direct molecular substitute for water.
Okay, how does that work?
The tricellose molecules interpose themselves between the water molecules, physically preventing them from aligning into that lethal crystalline lattice. Furthermore, the trehalose tightly binds to the external surfaces of the proteins and the lipid membranes, basically replacing the hydration shell.
So what you're saying is instead of the internal fluids freezing into sharp, jagged ice crystals, the extreme concentration of trehalose forces the liquid to transition into a highly viscous glass-like state.
Yes.
It achieves vitrification instead of crystallization.
Exactly. The cellular machinery is essentially suspended in a biological glass, perfectly preserving the structural integrity of the proteins in the membranes until the ambient temperature rises again.
That is brilliant.
It is, but managing the phase state of the water is really only one half of the cryobiological equation.
There's another half.
The other half involves the physical flexibility of the cell membranes themselves.
Oh, because the cell membrane isn't a static wall. It is a dynamic fluid envelope made of lipids fats. And fats react very poorly to cold temperatures.
They do. In freezing conditions, a standard lipid bilayer undergoes a phase transition. The lipid molecules pack tightly together, losing their fluidity and turning rigid.
And when that happens?
When the membrane solidifies, the transmembrane proteins, the channels that pump nutrients in and push waste out, are physically locked in place. Transport stops entirely. The cell starves and suffocates simultaneously.
The classic culinary analogy here is cooking fats, right?
Exactly.
If you have saturated fats, like butter or animal lard, the straight, rigid carbon chains pack neatly together, which is why they turn into a solid, hard block when you put them in the refrigerator.
Right.
But unsaturated fats, like olive oil, have slightly different chemical structures. They stay liquid even when chilled.
That different chemical structure is the key. Unsaturated fats have a cis double bond in their carbon chain. That double bond creates a physical 30-degree kink in the tail of the lipid molecule.
A kink.
Yes, and because of that kink, the lipids simply cannot pack tightly together no matter how cold it gets.
Okay, so how does the mushroom apply this? I mean, it can't really go to the store and swap its butter for olive oil when winter arrives.
No, it does something far more impressive. Genomic analysis of cold-adapted fungi reveals a high proliferation of genes encoding for a specific class of enzymes called fatty acid desaturases.
Desaturases. The name implies exactly what they do. They actively remove the saturation from the fats.
Yes. When the temperature drops, these enzymes are deployed directly into the cell membrane. They patrol the lipid bilayer, chemically attacking the straight saturated fatty acid chains. They intentionally strip away hydrogen atoms and force the formation of those cis double bonds.
Wait, they are actively installing kinks into the carbon chains of their own cellular walls.
In real time, it is a process known as homeoviscous adaptation. By artificially increasing the ratio of unsaturated to saturated lipids, the fungus maintains the exact required fluidity of its cell membranes, ensuring that nutrient transport continues flawlessly, even when the surrounding bog is encased in solid ice.
It is a stunning display of biochemical engineering. It vitrifies its internal fluids with tricellose to prevent ice crystals, and it actively desaturates its cellular walls to prevent lipid lockup.
It's a complete survival package.
Yeah. When you combine the cryobiology with the hollow structural engineering and the velutinal ester defense pathway, you are looking at an organism that has seemingly solved every environmental challenge the Orarctic can throw at it.
Seemingly.
Right, because evolutionary arms races are never stagnant. If you build a flawless, unbreachable chemical fortress, the ecosystem will simply evolve a specialized siege engine to crack it.
Which brings us to the consumers.
Yeah.
Because despite the searing dialdehydes and the impenetrable slime, the biomass of Lactarius trivialis is simply too valuable to remain completely unexploited.
Right. Calories are calories.
Exactly. Certain organisms have cracked the chemical code, and the most prominent of these is surprisingly large. We need to examine the reindeer paradox.
Yes. This is genuinely the most counterintuitive piece of data in our sources. Because we established that the stearoylvelutinal rapidly oxidizes into isovelleral and velleral, which are specifically designed to agonize mammalian pain receptors and trigger immediate rejection.
Yes, that is their whole purpose.
Yet in regions like Finnish Lapland, the northern milkcap is not just occasionally nibbled. It is a highly targeted critical food source for Reindeer tarandus, the local reindeer population. In the late summer and early autumn, these animals actively gorge on these mushrooms.
They absolutely devour them. The mushrooms provide an incredibly dense, easily digestible source of non-structural carbohydrates, primarily in the form of glycogen and those same sugar alcohols like mannitol and trehalose we discussed earlier.
Oh, the antifreeze sugars.
Exactly. The reindeer depend on this massive caloric influx to rapidly build up subdermal fat stores before the brutal Arctic winter sets in.
But the paradox is the mechanism of ingestion. How is a mammalian digestive system bypassing biological pepper spray without suffering catastrophic tissue damage to the mucosal lining of the throat and stomach?
The answer lies in thousands of years of sympatric coevolution. Reindeer and Lactarius have shared the same habitat for millennia. The evolutionary pressure for the reindeer to access that calorie source was immense.
So they evolved a workaround.
Right. While the exact salivary transcriptomes are still being mapped, the current consensus points towards specialized, highly advanced buffering mechanisms within the reindeer's rumen, the first chamber of their specialized stomach.
Wait, so what you're saying is the rumen has adapted specifically to neutralize the dialdehydes?
Yes. It is likely a combination of specialized salivary binding proteins that instantly coat and sequester the acrid sesquiterpenes before they can activate the pain receptors in the mouth, combined with a highly specialized microbiome within the rumen itself.
The gut bacteria do the heavy lifting.
Exactly. The bacteria in the reindeer's gut possess the specific enzymes required to rapidly degrade the toxic dialdehydes into harmless metabolic byproducts.
Yeah.
What is a searing, tissue-damaging toxin to a wolf or a human is dismantled and processed as premium fuel by the reindeer.
The ecological cascade of this interaction has to be profound. I mean, if the reindeer are actively hunting down and consuming massive quantities of Lactarius trivialis, they are directly altering the fungal biomass of the forest.
Oh, without a doubt.
And because this fungus operates symbiotically with the root systems of the trees, the reindeer are inadvertently shaping the entire subarctic flora.
You are absolutely right to look at the macroscopic cascade. Heavy reindeer grazing directly reduces the abundance of Lactarius fruiting bodies.
Yeah.
By disrupting the life cycle of the fungus, they alter the flow of nutrients in the soil.
Wow.
Research has shown that this top-down grazing pressure can actually serve as a buffer against shrubbification, which is the rapid, climate-driven expansion of woody shrubs across the warming tundra.
Oh, I've read about this.
Yeah, the shrubs rely heavily on these specific fungal networks to expand. So when the reindeer eat the fungi, they essentially starve the encroaching shrubs, maintaining the open tundra ecosystem.
So the reindeer crack the chemical lock, eat the fungus, and inadvertently keep the tundra from turning into a thicket. It's all connected.
But it's not just giant ruminants exploiting the mushroom. When we look at the insect specialists, the strategies shift from brute force chemical neutralization to, like, a surgical evasion.
The fruiting body of a large Lactarius is essentially an ecosystem unto itself. It's a high-rise condominium for specialized microfauna.
Like the columbella.
Yes. Consider the collembola, the primitive hexapods commonly known as springtails. They are tiny, highly mobile grazers, but they do not possess a massive multi-chambered rumen to detoxify the dialdehydes.
Right. If they trigger the velutinal ester pathway, they die instantly.
So how do they feed on it?
Evasion and precision, right?
Exactly. They have evolved highly specialized sensory organs on their antennae known as sensilla. These sensilla are exquisitely sensitive chemical detectors.
Okay.
As a collembola navigate the surface of the mushroom, they constantly tap the tissue, mapping the chemical concentrations. They use this sensory data to locate specific, safe micropockets of undefended tissue.
Like what? Such as mature spores or degrading hyphal threads. They feed surgically without ever rupturing the pressurized lactifers that house the binary weapon.
They are essentially using chemical radar to tiptoe through the minefield, extracting calories without tripping the wire. That is elegant.
Very elegant.
But then you have the pleasing fungus beetles, the family Erytilidae. They take a completely opposite, far more aggressive approach.
They do. The Erytilidae larvae do not evade the toxins. They fully immerse themselves in them. These larvae are obligate mycophages, meaning they only eat fungi. The adult beetles lay their eggs directly into the flesh of the Lactarius.
Right.
And when the larvae hatch, they begin to blindly tunnel through the internal tissue.
But tunneling requires chewing, which means they are constantly rupturing the cells and triggering the dialdehyde cascade. They are bathing in the biological pepper spray.
They possess their own internal enzymatic resistance to the toxins.
Yeah.
But the brilliance of their strategy is how they weaponize the mushroom's defense against the rest of the forest.
Oh, I see where this is going.
By living deep inside the highly toxic, acrid flesh of the Lactarius, the larvae are entirely shielded from predatory birds, parasitic wasps, and predatory ground beetles.
Because the predators avoid the mushroom.
Exactly. The predators recognize the mushroom as toxic and avoid it entirely, inadvertently ignoring the fat, nutrient-rich beetle larvae feeding safely inside.
They use the chemical fortress as a private panic room. The fungus built the walls, and the beetles just moved in.
Precisely.
But the most conceptually bizarre insect interaction in our sources belongs to the Aratidae, the flat bugs. Their anatomical adaptation reads like science fiction.
It really does. The flat bugs bypass the macroscopic defense systems entirely by operating exclusively on a microscopic cellular level. They do not possess mandibles. They cannot chew or crush the fungal tissue.
Which means they rarely trigger a massive localized rupture of the lactifers. So instead of jaws, they have a stylet, a highly specialized hypodermic feeding tube. And the morphological adaptation here is absurd. In certain species of Aratidae, this coiled stylet is actually longer than the entire body of the insect.
It is a remarkable piece of micromachinery. When the flat bug locates a suitable fungal hypha, a single, microscopic thread of the mycelial network, it uncoils this massive stylet. The tip is incredibly sharp, allowing it to pierce directly through the tough, chitinous outer wall of a single fungal cell.
Wow. And once it pierces the cell wall, it basically acts like a microcatheter.
Right.
It precisely extracts the nutrient-rich liquid cytoplasm from inside the cell, leaving the surrounding tissue almost entirely undisturbed. The macroscopic chemical defense mechanisms of the mushroom simply don't register the attack because the damage profile is just too incredibly small.
It is the ultimate surgical strike. The flat bug drains the metabolic lifeblood of the fungus one microscopic cell at a time.
Unbelievable. So we have charted the interactions from the top down. We've seen how the reindeer use massive enzymatic power to crush the defenses, how the beetles hide inside them, and how the flat bugs surgically bypass them. We have looked deeply into what eats the mushroom.
Yes.
But this brings us to the true paradigm shift of this deep dive. The most vital, globally significant research regarding Lactarius trivialis isn't about what eats the mushroom. It is about what the mushroom eats.
Yes.
And how its voracious appetite dictates the very chemistry of the earth. So let's dig into the 2025 Patchett study.
This represents the absolute frontier of modern soil ecology. This is where we stop looking at Lactarius trivialis as a discrete individual organism and start analyzing it as a master regulator of the terrestrial ecosystem.
Right.
Lactarius trivialis is an ectomycorrhizal fungus, commonly abbreviated as ECM. This classification is the key to everything it does.
Okay, ectomycorrhizal. Let's break that down for clarity. It means the fungus doesn't exist in isolation. Its subterranean body, the massive sprawling network of microscopic mycelial threads, physically merges with the root systems of the dominant flora.
Specifically, the cold-adapted spruce and birch trees of the Oro-Arctic.
Right.
Yes. It forms a symbiotic mantle around the tips of the tree roots. Now, the textbook understanding of this symbiosis is a straightforward economic exchange.
Quid pro quo.
Exactly. The tree, operating high up in the canopy, has access to sunlight and carbon dioxide. It performs photosynthesis, creating energy-rich sugars, and pumps a significant percentage of those sugars down into its roots to feed the fungus.
And in return, the fungus, which has a massive surface area distributed throughout the soil, acts as an incredibly efficient biological mining network.
Right.
It absorbs water and essential trace minerals like phosphorus and nitrogen and pipes them back up into the tree roots.
That is the established paradigm. But the 2025 study, published by Patchett and colleagues, completely rewrote our understanding of the sheer aggression and dominance with which ECM fungi, particularly Lactarius and related species, manage this economy in harsh, nutrient-scarce environments.
They focus specifically on the nitrogen cycle in cold boreal soils, right?
They did. Historically, the assumption was that the nitrogen cycle in a freezing bog was passive and sluggish. Because cold temperatures generally suppress chemical reaction rates, scientists assumed the breakdown of organic matter and the subsequent release of nitrogen was a slow, ambient process.
Yeah, that makes sense. Things rot slowly in the cold.
That was the consensus. But Patchett's team introduced rigorous isotopic tracing methodologies. They utilized soil cores injected with heavy nitrogen isotopes 15N to trap the exact flow and processing speed of nitrogen in plots dominated by ECM fungi versus plots where the fungi were absent or suppressed.
And the data they recovered was shocking.
It was.
They discovered that in ecosystems heavily colonized by ECM fungi, like L. trivialis, the rate of gross nitrogen mineralization increased by an astonishing 73%.
Yes, 73%.
Wait, 73%. That is a massive statistical anomaly. That isn't a slight optimization. That is the fungal network fundamentally hijacking the chemical cycle of the forest floor and throwing it into absolute hyperdrive.
If we connect this to the bigger picture, it implies extreme active intervention. The fungus is not passively waiting for nitrogen to naturally detach from decaying leaves and slowly drift into its mycelial net. It is actively hunting.
Hunting for nitrogen.
Yes. The mycelium secretes highly specialized proprietary oxidative enzymes directly into the surrounding soil matrix. These enzymes are designed to chemically attack and depolymerize the incredibly tough recalcitrant organic molecules locked inside deadwood, fallen needles, and decaying moss.
So it is aggressively breaking down the complex polymers to strip mine the organic nitrogen locked inside. It hoards this vital element and funnels it directly to its symbiotic host trees.
Yes. But the revelation isn't just that it feeds the trees. The revelation is the ecological consequence of its hoarding.
Okay, what happens when it hoards the nitrogen?
By aggressively stripping the nitrogen out of the soil matrix, Lactarius trivialis fundamentally starves the rest of the microbial community. It triggers intense subterranean competition, drastically outcompeting the free-living saprotrophs.
Okay, the saprotrophs. Those are the standard decomposers, the ambient bacteria and non-symbiotic fungi whose entire job is to rot dead plant matter.
Exactly. Saprotrophs require a baseline level of ambient nitrogen to produce the enzymes necessary to break down dead leaves and wood.
Right.
But the Lactarius network is so massive, so pervasive, and so efficient at extracting nitrogen that the ambient levels plummet. The saprotrophs are utterly starved of the raw materials they need to function.
So what does this all mean? By dominating the nitrogen market, the mushroom effectively shuts down the primary rot makers of the forest. This creates a massive bottleneck in the decay cycle.
It does.
And this phenomenon actually has a name in the literature, right?
It does. It is referred to as the Gadgil effect, named after the ecologist Ruth Gadgil, who first hypothesized the mechanism in the 1970s. But the 2025 Patchett data provided unprecedented empirical validation of its sheer scale in the Oro-Arctic.
Wow.
Because the saprotrophs are starved and suppressed, the overall decomposition rate of the forest floor slows to a crawl. The dead needles, the fallen branches, the moss, it simply stops rotting at a normal rate.
And if it stops rotting, we have to look at the massive planetary implication here.
Absolutely.
The physical structure of a dead spruce branch is composed primarily of carbon drawn from the atmosphere. When a branch rots, the decomposers break those bonds and release that carbon back into the atmosphere as CO2. But if Lactarius trivialis is starving the decomposers,
then the carbon is never released. The organic matter simply accumulates on the forest floor layer upon layer year after year, eventually compressing into peat and dense soil organic matter.
So the fungus is trapping the carbon. By relentlessly hoarding nitrogen to feed a few spruce trees, this slimy, unassuming mushroom is inadvertently locking millions of tons of carbon into the boreal soils. It is acting as a massive subterranean planetary thermostat, actively suppressing the release of greenhouse gases.
It completely reframes our perception of the organism. It is not merely existing in the landscape. Its metabolic processes are a load-bearing pillar of the global carbon cycle. The sheer scale of the carbon sequestered by these ECM networks in the northern hemisphere is staggering. It is a vital buffer against runaway atmospheric heating.
It is genuinely awe-inspiring. We start by looking at a hollow stem and a chemical defense mechanism, and we end up uncovering a planetary regulatory system.
It's a huge leap.
It really is. Now, given the sheer majesty of what we just discussed, an organism that manipulates the global climate, deploys searing chemical dialdehydes to burn the mouth of its enemies, and synthesizes antifreeze to survive sub-zero bogs, it seems absolutely absurd that humans walk into the forest, look at this exact organism, and think, you know what, I should chop that up and put it in a salad.
It does seem highly counterintuitive.
Totally crazy.
Yet, the human interaction with Lactarius trivialis is a massive part of its story. In Finland, Estonia, and across the Baltic states, the hakarusku is not viewed as a toxic biological weapon. It is one of the most commercially important, culturally revered, and aggressively foraged wild foods in the entire region. It is a cornerstone of their culinary heritage.
But the biochemistry hasn't changed. We just spent 20 minutes establishing that if a mammal bites into this mushroom, the velutinal ester pathway triggers, and the mouth is flooded with searing cytotoxic dialdehydes.
Right.
If a human eats this raw, they do not get a pleasant peppery tingle. They face a severe medical reaction.
Absolutely. Ingestion of raw Lactarius trivialis results in severe, immediate digestive distress. We're talking about intense nausea, violent cramping, and an overwhelming, lingering burning sensation throughout the entire gastrointestinal tract. The raw tissue is violently defended.
So how are the Finnish foragers getting around this? We clearly haven't evolved a highly specialized multi-chambered rumen like the reindeer to enzymatically degrade the toxins.
No, we have not bypassed the defense biologically, we bypass it technologically.
Okay.
The entire culinary tradition revolves around a highly specific, non-negotiable preparation method known in Finnish as riapes.
Riapes. The linguistic texture of that word perfectly matches the action. What is the physical process?
It essentially translates to aggressive parboiling or blanching. To render the northern milkcap safe for human consumption, you must submerge the freshly harvested mushrooms in a large volume of rapidly boiling water for a minimum of 5 to 10 minutes.
And this application of extreme, sustained thermal energy fundamentally alters the biochemistry of the fungus in two distinct ways, right?
Yes,
The first has to be thermal degradation of the dialdehydes.
precisely. The highly reactive sesquiterpene dialdehydes, the isovelleral and velleral that cause all the pain, are structurally unstable at high temperatures.
So the heat breaks them down.
Right. The boiling water physically cleaves the reactive aldehyde groups from the molecular backbone, fundamentally destroying their ability to interact with mammalian pain receptors. You are chemically disarming the landmine.
And what's the second mechanism?
Solubilization. Even as the compounds degrade, the remnants and any unreacted precursors are highly water-soluble. As the cellular structure of the mushroom relaxes in the heat, these toxic compounds leach completely out of the fungal tissue and dissolve directly into the boiling water.
Which means the water left in the pot is no longer just water. It is a highly concentrated cytotoxic soup of extracted fungal defense chemicals.
It is entirely toxic. The absolute most critical step of riapes is that you must immediately drain the mushrooms and completely discard that boiling water. You must never use it for a broth or a stock.
Right, that would be a disaster.
However, in a brilliant display of rural resourcefulness, many traditional foragers will collect that toxic water, allow it to cool, and apply it directly to their agricultural gardens as a highly effective, organic, broad-spectrum pesticide.
That is incredible. They extract the defense mechanism of the fungus and redeploy it to protect their cabbages from aphids.
Exactly.
But, from a purely culinary standpoint, I have to question the texture. You take a highly viscid, water-filled mushroom and aggressively boil it for 10 minutes. Doesn't the structural integrity completely collapse? Doesn't it just turn into a gray, unappetizing mush?
This is where the biomechanics of the fungal cell wall shine. Fungal cell walls are built from chitin, the same tough biopolymer that makes up the exoskeletons of insects.
Oh, right.
Unlike plant cellulose, which breaks down quickly in boiling water, the chitin matrix of L. trivialis is incredibly robust.
Right.
After the riapes process is complete, you are left with a surprisingly firm, highly resilient, and delightfully chewy mushroom that possesses a deep, savory, earthy flavor profile.
And historically, this isn't a food they eat immediately, right? The caloric value in a subarctic winter is far too high to just consume all at once. The harvest must be preserved.
Preservation is the primary goal. Once boiled and cooled, the traditional method involves layering the mushrooms heavily in coarse salt, packing them tightly into wooden barrels or glass jars.
Oh, salt packing.
Yes, the extreme osmotic pressure created by the salt draws out any remaining internal moisture, creating a completely inhospitable environment for any residual bacteria or mold. Properly salted, these mushrooms can remain perfectly viable in a root cellar for months, providing a vital source of complex carbohydrates and trace minerals throughout the deep winter.
But when you actually want to eat them in the middle of January, you can't just eat heavily salted, preserved fungus. You have to reverse the process.
You do. You remove the required portion from the salt barrel and soak them in multiple changes of fresh, cold water overnight. This reverses the osmotic gradient, drawing the excess salt out and rehydrating the flesh.
And from there, the culinary applications within Baltic cuisine are pretty expansive.
Oh, very much so.
But the most iconic dish has to be cinesalati.
Without question. It is the traditional Finnish Christmas mushroom salad. The rehydrated mushrooms are finely chopped and folded into a rich mixture of freshly whipped cream, sharply diced raw onions, and perhaps a touch of white pepper or vinegar.
Wow, that sounds intense.
It is a heavily textured, savory, umami-rich dish that is deeply tethered to the geography of the region. But beyond the traditional salad, its firm texture makes it highly versatile. It is commonly folded into heavy meatloaves to retain internal moisture, baked into savory root vegetable pastries, and it has even found a massive secondary market as a premium topping in modern pizzerias across Helsinki.
It is a remarkable journey from a toxic bog dweller to a commercial pizza topping.
It really is.
But anytime we discuss the commercial or personal foraging of wild mushrooms, we have to address the inherent profound risks. The wild is not a grocery store, and Lactarius trivialis shares its habitat with several closely related species.
Yes, you have to be extremely careful.
Because if you are applying the Riapes technique to the wrong organism, the results range from ruined dinners to severe medical emergencies. We really need to detail the lookalikes.
Accuracy is paramount in taxonomy, and it is a matter of safety and foraging. Let's start with the benign relatives. The most common confusion occurs with Lactarius utilis, known as the pallid northern milkcap.
Right. And from the literature, L. utilis is genetically adjacent to trivialis and is entirely edible if prepared correctly. But the morphological differences are distinct, if you know what to look for.
The primary divergence is coloration and mucilage. L. utilis lacks the deep leaden gray or purplish hues of the true northern milkcap. It presents with a much paler, almost washed-out leather or pale beige coloration.
Okay.
Furthermore, while trivialis is slimy, utilis is extraordinarily viscid. The mucilage layer is so thick and tenacious that the cap acts as an aggressive adhesive, collecting a dense, impossible-to-clean layer of pine needles, dirt, and dead insects. It is safe to eat, but the sheer labor required to clean the cap often deters commercial foragers.
Then we have Lactarius uvitis, the purple staining milkcap.
Also edible following the rigorous boiling process. But the chemical markers are fundamentally different here.
How so?
When we discussed trivialis, we noted that the bright white latex oxidizes into a grayish-green crust. With L. uvitis, the latex reacts entirely differently to oxygen. When the flesh is bruised or the gills are snapped, the latex aggressively stains the surrounding tissue, a vivid, deep lilac or purple color.
Oh, wow.
It is a stunning visual reaction, but it clearly differentiates it from our target species. Similarly, there is the gray milkcap, Lactarius viridis, which is physically much smaller, more fragile, and its latex oxidizes into a flat, solid gray, completely lacking the green hues.
Okay, so those are the safe misidentifications. If you make a mistake and end up with utilis or uvitis and you follow the boiling protocol, you are physically fine.
Exactly.
But there is an imposter species occupying the exact same bog ecosystem that presents a genuine threat. We must explicitly clarify the danger of the fenugreek milkcap, Lactarius helvis.
This is where reliance on common names becomes incredibly dangerous. It's called a milkcap, but this is a profound misnomer. Lactarius helvis does not produce the characteristic milky latex that defines the genus.
Right, it doesn't bleed milk.
No, if you snap the stem or slice the gills of the fenugreek milkcap, it exudes a completely transparent, watery fluid.
Okay, that is the ultimate red flag. If it doesn't bleed milk, it is not the northern milkcap.
Right.
And beyond the visual marker, the olfactory signature is entirely different too.
It is dominated by a completely different chemical profile. As the fruiting body of Lactarius helvis matures, and particularly if it begins to dry, it synthesizes a volatile compound called sotalon. Sotalon emits a highly aggressive, sweet, aromatic odor that smells remarkably similar to strong licorice or maple syrup.
So you have a mushroom that bleeds clear water and smells intensely of licorice. And why is identifying this specific imposter so critical?
Because the chemical defense system of L. helvis relies on a totally different suite of toxins than the dialdehydes found in trivialis. And crucially, these specific toxins are incredibly thermally stable. They do not degrade under extreme heat.
Wait, so what you're saying is the boiling protocol, the ryopase, does absolutely nothing to disarm this mushroom?
It does nothing. If a forager accidentally includes Lactarius helvis in their harvest, meticulously boils it for 10 minutes, salts it, and consumes it months later, they will suffer severe, debilitating gastrointestinal poisoning. The toxins persist entirely through the preparation process.
Yikes. So we have established absolute non-negotiable rules for the field. If the stem is in a perfectly hollow, biomechanically optimized cylinder, and if the latex doesn't emerge bright white and slowly oxidize to a grayish green, you just leave it in the bog.
Those are the uncompromising structural and chemical metrics. You must verify the engineering before you attempt the harvest.
Well, this has been an absolutely staggering exploration. We began in the 19th century scientific literature, watching Elias Magnus Fry's attempt to decode the evolutionary history of an organism based purely on its peppery taste and milky exudate.
Yeah, amazing history.
We examined the flawless biomechanics of its hollow stem, designed to maximize structural integrity while minimizing mass in a waterlogged bog. We mapped the violent, instantaneous enzymatic cascade of the velutinal ester pathway, analyzing how the rupture of cellular compartments synthesizes searing dialdehydes to deter predators, sterilize wounds, and deploy antioxidant defenses.
And we charted its cryobiological mastery, detailing how it floods its cytoplasm with trachelose to vitrify its internal fluids and actively desaturates its lipid membranes to maintain cellular transport in sub-zero environments. We explored the complex web of consumers, from the heavy biome-altering grazing of the reindeer to the surgical cellular extraction performed by the microstylates of the flat bugs.
And perhaps most significantly, we looked at the 2025 Patchett data, revealing how this single organism acts as a planetary regulator. By aggressively mining the soil for nitrogen, it starves the free-living decomposers, fundamentally slowing the decay cycle of the Arctic and locking millions of tons of carbon securely into the earth.
It's incredible.
It is an organism that simultaneously sustains ancient human culinary traditions and fights modern climate change. But before we conclude, the sources indicate one final emerging avenue of research that suggests this fungus might have one more massive trick up its sleeve.
It does, and it returns us precisely to its common name, the Slimy Lead Lactarius.
Right.
For centuries, the lead simply referred to the dull, purplish-gray coloration of the juvenile cap. But cutting-edge biochemical assays are beginning to take that nomenclature entirely literally. The research is shifting toward the phenomenon of heavy metal sequestration.
Heavy metal sequestration. Are you saying the fungus is actively pulling toxic industrial metals out of the soil matrix?
We are discovering that several closely related species within the broader Russulaceae family possess a remarkable, largely unexplained trait. They are hypoaccumulators of heavy metals.
Wow.
Their mycelial networks possess the physiological capacity to absorb highly toxic, non-biological elements, specifically silver, lead, and mercury, directly from the contaminated soil, transport them up the mycelial network, and safely sequester them deep within the tightly bound tissues of the fruiting body.
Without poisoning themselves in the process.
Exactly. They likely utilize specialized chelating proteins like metallothioneins to bind the toxic metal ions, effectively neutralizing their reactivity and storing them inertly within the cellular vacuoles.
That is wild to think about.
It is. Given the intense metabolic similarities across the genus, soil ecologists in 2025 and 2026 are actively funding field studies to determine the exact sequestration capacity of Lactarius trivialis. They are investigating whether this massive, ubiquitous fungal network is currently functioning as a silent subterranean filter, actively pulling decades of human industrial pollution, the lead, the mercury, the airborne particulates that have settled into the northern bogs out of the oral Arctic ecosystem.
So an organism that is already a cornerstone of the Arctic food web, a master of chemical defense, and a massive carbon sink regulating the global climate might also be our most powerful natural ally in large-scale bioremediation.
It is very possible.
It might literally be mining the lead out of our poisoned earth. That just fundamentally changes how you interact with the wild. The next time you find yourself off the trail, standing in a damp, freezing, mosquito-filled bog, look past the towering trees. Look down at the mud, the moss, and the slime, because the entities that are truly engineering the planet, trapping the carbon, and potentially cleaning up our deepest industrial mistakes are hiding right beneath your boots.
They really are.
Thank you for joining us on this deep dive. Keep questioning the architecture around you, and we will see you next time.